only medium through which we can communicate. Yes, doctor, very good. Perfect. I'm waiting for everyone to reply because no, everyone has to reply in the chat box. This is something which is very much compulsory in my lectures. Chalo, Kavita Bishnoi, very good, beta. So, okay, Muskan, very good. Just give me 1 minute. Okay. Hmm. Okay, Dr. Ishwar, fine. So let's start with the class, guys. So we are going to start with the carbohydrates, but before that, let me introduce um to this particular app, that is the Oracle. And yes, guys, so we will be covering the basics of carbohydrates in this particular session. But before that, a little bit about the app, guys, that this particular app, we have certain no features of this particular app. So over here, we have the live classes, the recorded classes, the notes, and the MCQs, right? So all the features, no live classes, which I'm taking right now, it's one like live interaction with you people, right? So beyond that, we have the recorded classes, which is available on this particular app. All the notes, they are being designed very curatively, right? According to the examination pattern, and also for the upcoming no these NEET PG and FMG examinations. Beyond that, we have the MCQ session also, right? All the important MCQs, the previous year questions, the previous year topics are all covered in this particular app, right? So yes, even if you guys want to purchase this particular app, you can use a discount code, that is Dr. Abhijeet 20, right? You'll automatically get a discount, and you will be having access to everything which is mentioned on this particular screen. So a basic 3-month plan is there, right? Which is of rupees 15,000, standard 6-month plan, and we have mentorship plan of 12 months, right? So yes. Hmm. So this is the no, you can download it from Google or Google Play Store or like the Apple Store, right? And everywhere, no, you can get an access for the latest updates and everything. You can follow the Facebook, Telegram, and the Instagram pages as mentioned on the screen, right? So yes. So without wasting much more time, let us just start with today's session on introduction to biochemistry or the biochemistry of carbohydrates. So guys, have you studied biochemistry of carbohydrates before? So just let me know, guys, if you have studied it before or not. So yes, the only medium to communicate is via chat box, guys. So yes. Hmm. Let us now start with the biochemistry of carbohydrates, the part one, which I will be discussing right now. So now, guys, what is the definition of a carbohydrate? What is the definition of a carbohydrate? So remember, guys, the carbohydrates are nothing but the hydrates of carbon. They are nothing but the hydrates of carbon, right? Hydrates of carbon. Now, the general formula for carbohydrates, or if I can say monosaccharide, it is nothing but CnH2ON. Basically, N denotes the number of carbon atoms. Okay, just a moment. Guys, are you able to see the notes? Okay. I think now it's visible, Kavita. Very good. What about rest, guys? Is it visible? Muskan, doctor, what about the rest? Students, kindly reply in the chat box, guys. I hope it is visible now. Yes. Chal. Muskan, very good. Very good. Very good. Doctor, very good. When I reply karte rehna, just try to reply in the chat box so that we can have one-to-one interaction, right? Otherwise, it will be a very boring class, right? So it is a very no general formula where the carbohydrates are basically composed of carbon, hydrogen, and oxygen, the three which you can see, and N basically denotes the number of carbon atoms. N denotes the number of carbon atoms, right? Now this is the general formula, guys, right, for carbohydrates, or I can say the monosaccharides. The general formula, okay, for monosaccharides. Mono means single, saccharides means sugar. Carbohydrates are also called as sugar, right? Now, the definition which you have to write in the exam, guys, let us just try to focus on that. Yes. So remember that the carbohydrates are aldehyde. Carbohydrates are aldehyde. Now, can anyone tell me in the chat box what is the functional group? Aldehyde, it is denoted by which functional group? Yeah. Okay. Anyone? The functional group. Yes, doctor, very good. It is CHO. It is CHO. Very good, Muskan. Very good. Very good, Peter. It is CHO. So carbohydrates are aldehyde, that is CHO, or I can also write it like this, C double bond O and H like this, right? So it is nothing but the CHO. Carbohydrates are aldehyde, right? These are aldehyde or keto derivatives, right? These are keto derivatives. Now, when I say keto derivatives, guys, can anyone tell me the functional group for keto derivatives? Right. Yes, I'm waiting for your answers, guys, because you all have to answer me. The keto derivatives. Yes, it is C double bond O, right? It is C double bond O, right? Yes, Muskan, very good, beta, very good. So carbohydrates are aldehyde or keto derivatives of polyhydroxy alcohol. Very, very important. It is polyhydroxy alcohol. So poly means many, hydroxy means OH group. OH group. So let us just try to understand this particular definition on the next slide with the help of a schematic representation, guys. So again, guys, you have to write the notes along with me, right? You have to write the notes along with me, right? And I hope everyone is writing, listening to my lecture. I hope the copy and pens are with you, right? So yes, I hope everyone has written this particular slide, guys. Just give me a thumbs up if you have written this. So this is the definition that has to be known in the exam, right? So I'm just waiting for your thumbs up, guys, because yeah. Chalo. Very good. Just give me a thumbs up, guys, if it is done. Now, let us just see the formation of the carbohydrates, guys, over here. So you, according to the definition, we have something called as the polyhydroxy alcohol, right? We have polyhydroxy alcohol. Now, in this polyhydroxy alcohol, we can take one example that is called as the glycerol, the very basic example that is called as the glycerol. Right? Now, when I say hydroxy, hydroxy means the OH group. Poly means many. Poly means many. More than one hydroxyl group. More than one hydroxyl group. So let us just draw this, guys. So we know we have the 3-carbon compound, right? Glycerol is a 3-carbon compound, and we have more than one OH group. So we have 3OH, 3OH, right? Let it be the first carbon, the second carbon, and the third carbon. So, and okay, now guys, in the chat box, just try to answer this, guys. Just try to answer this. Okay, carbon ki valency kya hoti hai? What is the valency of carbon? carbon will make how many bonds to know complete its outer shell? Very good, Muskan. Perfect, guys. So you already know. Let us. If we talk about the first carbon, it already has two, right? So automatically, I have to add two more H over here, right? CH2. The second carbon already it has three bonds, right? To fulfill, I have to add one more H, and at the last, yes, we have to add two hydrogen, right? So yes, again, the third carbon will have four valency, one above, two sideways, and one this, right? So automatically, this is something which is called as the polyhydroxy alcohol, or it is the glycerol. Now, from this, guys, from this, basically the carbohydrates are formed. Now, from the first carbon, guys, what we are trying to do, we are trying to remove one hydrogen over here and one hydrogen from this. So automatically it will be minus H2. Okay. So in the first carbon, guys, only oxygen will be left, and oxygen is always bonded like double bond, and we have one H left like this. Or just make it as per your convenience, guys. This is, yeah, this. So one H gone from this side and one H. So C double bond O. So you can anyway see the carbon has fulfilled all the four valencies like this. So this is an aldehyde derivative. This is an aldehyde derivative. Rest the entire structure remains the same, right? Rest the entire structure remains the same. So this is something which is called as yes. So from the glycerol, okay, we are adding an aldehyde group. So this structure it is called as glyceraldehyde, right? This is called as glyceraldehyde. This is called as glyceraldehyde, right? Now again, guys. So yeah, the next one, guys. You already know. From the second carbon, we will try to remove one hydrogen from here and one hydrogen from here. So there will be from the second carbon, right? From the second carbon, we are removing two hydrogens. So again, the structure is like this. Now this carbon is left alone with the oxygen. So automatically it will share double bond to complete the valency. Then we have CH2OH. This is the keto derivative, right? This is the keto derivative, and it is also known as dihydroxy acetone. It is also known as dihydroxy acetone. So remember, guys, over here that, okay, now beta, this structure, yes, let me just try to draw like this. Okay. Now remember, guys, that this structure, it is the structure of acetone. And acetone ke first carbon, on the first carbon and the third carbon of acetone, we have OH group. So it is dihydroxy acetone. It is dihydroxy acetone, guys. It is dihydroxy acetone. So again, guys, we have the aldehyde derivative, and we have keto derivative. So again, going to the definition, carbohydrates are aldehyde or keto derivatives of polyhydroxy alcohol. So I hope it is clear now. Polyhydroxy alcohol means the glycerol, the simplest example that we took, and how the carbohydrates are formed. Basically, these are aldehyde or keto derivatives, right? See, guys, one more important thing that I want to say, the aldehyde group, it is always present at the first carbon atom, right? The keto group, it is always present at the second carbon atom, right? So the keto group, it is always at the second carbon, and the first carbon contains the aldehyde group. So I hope this is clear to you guys. Just give me a thumbs up in the chat box, guys, if this particular picture is clear to everyone. चलो फटाफट से एक थम्स अप दे दो जल्दी से. I hope you all are writing along with me, guys. I'm waiting for your yes, doctor. Very good. I'm waiting for everyone to reply in the chat box, guys. Yes. Doctor, very good. I hope, Doctor, you're writing. Kavita Bishnoi, very good, Peter, very good. Now, what about rest, guys? I hope you all have written. You're writing along with me. Hmm. Sarath, Muskan. Okay. Sure. Sure. So that is how, see if the definition comes. If you just write the simple definition and along with that, if you just draw it, it will be like a see the answer should be presentable. No one is asking you to write thesis, right, in the exam. It should be point to point answer. It should be schematic. It should be no, yeah, short, concise, point to point answer, and it should be like well illustrated. So now coming on to three questions that can be asked in the Viva also, right? The three questions that can be asked in the Viva exam also. Guys, remember that the parent alcohol, the parent alcohol, it is called as the glycerol, guys. Okay. The parent alcohol, it is called as the glycerol because it is the one through which we are forming the carbohydrates. Now, guys, apart from that, the second question, the simplest carbohydrate, guys. Now, we have two over here. The two carbohydrates are formed. One, it is glyceraldehyde. From the glycerol, they are only formed. We have glyceraldehyde. And the second one, guys, it is dihydroxyacetone. Dihydroxyacetone, right? Dihydroxyacetone. And out of this, guys, remember the glyceraldehyde is being the parent carbohydrate. Okay, it is the parent carbohydrate, right? It is the parent carbohydrate. So these are the 3 points that you guys have to remember. Now coming on to the next part, guys. I hope you all have written this, guys. Just give me a thumbs up in the chat box. Continuously, you have to reply, guys, right? So just give me a thumbs up in the chat box. You understood the basic thing, right? Now there is something called as symmetric and asymmetric carbon atoms. When I will be discussing the isomerism, I will be focusing on that. Symmetrical means basically the functional groups are same, the carbon, okay, all the functional groups remains the same, whereas the asymmetrical part, it is either 2 or 3 or 4, all the entire no, through the carbon is bonding to it is entirely different. So right now I hope this is clear, guys, everyone. So yes, I hope it is clear, guys. Just give me a thumbs up if it is done, right? Sure. Now coming on to the next one, that is the classification of the carbohydrates. That is the classification of the carbohydrates. Kavita, very good. Very good, Muskan, very good. Yes, I want every one of you to reply, right? Now, classification of carbohydrates. So the carbohydrates are basically divided into 4 categories, I would say. Yes, we'll draw a very simple flowchart. I'll try to make it very, very simple for you guys, right? Yeah. We have the very first one, guys, that is the monosaccharides. Monosaccharides. Then the second one, we have disaccharides. Okay. Let us just write down one by one. Okay. The second one, it is disaccharides, polysaccharides, right? And oligosaccharides. Yes. Oligosaccharides. Now, when I say mono, guys, just by the name only, mono means single and saccharides means sugar. Okay, so we have single sugar unit, right? We have single sugar unit, right? Single means one sugar unit. Now when I say di, guys, di means 2 sugar units, right? Or it is 2 potential sugar units. 2 potential sugar units, right? Polysaccharides, basically it is poly means many. Many means more than 10 sugar units, more than 10 sugar units. And oligosaccharides, basically it is, okay, we can just say it as the few sugar units, basically ranging between 10, 3 to 10 sugar units. So we have monosaccharides, disaccharides, polysaccharides, and oligosaccharides. So we will be covering one by one each category, mono, di, and poly. So first I will be focusing on the monosaccharides. First I will be focusing on the monosaccharides. Now, so before coming to this flow chart, let us first try to understand this in a very simple manner. Okay. Okay. Now, guys, the very first thing that I want to tell you, it is the general formula for monosaccharides. We have already discussed this general formula for monosaccharides, and it is nothing but CnH2ON, where denotes the number of the carbon atoms. N denotes the number of carbon atoms, right? Now, when we say monosaccharides, guys, the definition automatically says these are the sugars, okay? The sugars which cannot be hydrolyzed further, which can't be hydrolyzed further. They cannot be broken down by addition of water, right? These are the simplest sugars, guys, because they cannot be broken down. Sugars which can't be hydrolyzed further. Okay. Now, when I say monosaccharides, basically it means that we have the one sugar unit. We have one sugar unit. Okay. And remember, the monosaccharides are nothing but they are the building blocks of all carbohydrates. They are the building blocks of all carbohydrates, right? So they are the building block of all carbohydrates. Now, now these monosaccharides, I hope everyone has written up till here. Guys, just give me a thumbs up in the chat box that you have written up till here. No. Yes, just give me a thumbs up in the chat box, guys, if you have written up till here. Yeah, I'm waiting. Yes, doctor, very good. What about rest, guys? What about rest? Are you all present here? Yes, just let me know. Hmm. Sure. Muskan, yes. What about Kavita? Have you all written this? Yes, Kavita, very good. Very good. Very good. Perfect. Perfect, guys. Chal. Let's move on to the classification of monosaccharides, guys. Now, monosaccharides basically they are classified okay, depending on the number of carbon atoms. Depending on the number of the carbon atoms, guys, right? So it can be 3, 4, 5, and 6. Now, you guys already know minimum requirement is 3. We have 3 carbon atoms. We have 4 carbon atoms, 5 carbon atoms, and 6 carbon atoms. Now, depending on the number of carbon atoms, so we have some general names, right? We have some general names, right? And we have two formations, guys, over here. Either the carbohydrate which will be formed, either they are aldehyde derivatives, so these are called as aldoses, or either they are keto derivatives, so they are called as ketoses. Now, when I say aldoses, guys, you already know it is an aldehyde derivative, so it will contain a functional group CHO. And ketosis, it is C double bond O. The keto group, it is always a carbon, and the aldehyde group, it is always present at the first carbon. Now, guys, let me show you a sample, guys, from the notes of the Auricle. They have finally curated this in a very simpler manner. The whole flowchart which you can see on the screen. Now, this is from the Auricle app, right? All the notes are being curated like this in a very no very concise, very crisp, and in a presentable form, like in the form of flowcharts, right? To the point answers are there. So when we say three carbon atoms, beta, when we say three carbon atoms, we have triosis, four carbon atoms, tetrosis, then five carbon atoms, pentosis, then six carbon atoms, we have hexosis, and seven, we have heptosis, right? So depending on upon the number of the carbon atoms, we have different monosaccharides over here, right? Triose means three carbon atoms, three carbon atoms. Tetrosis means four carbon atoms. This is one of the sample which I'm showing from the notes. So automatically the notes are highly curated like this. You will be able to access to the notes once you download this particular app, and you just go to the biochemistry part. You will be able to access all the notes. So they are very beautifully curated. Then we have five carbon atoms in pentosis, six carbon atoms in hexosis, and heptosis means seven carbon atoms. Seven carbon atoms, right? Now coming on to the general names. Coming on to the general names, which we have already discussed over there. So the three carbon atoms, they are called as triosis, right? The four carbon atoms, these are called as tetrosis, right? Then the five carbon atoms, it is called as pentosis, right? And six carbon atoms, it is called as hexosis, right? Hexosis, right? We have seven heptosis bolenge, right? So automatically we are just focusing over here. Now, teen carbon atom mein. Okay, now I want answers from you guys without looking. So if an aldehyde group, it is attached to the first carbon, let us suppose these are the three carbon atoms. If an aldehyde group is attached to the first carbon, what will be the name of the aldoses, guys? We have just discussed. What is the aldehyde derivative of the three carbon atoms, right? Polyhydroxy alcohol. What is the aldehyde derivative? What is the name, guys? Chalo, I want the answers from everyone over here. फटाफट से बता दो जल्दी से. Yes, Muskan, very good, beta. We just discussed कि तीन carbon atoms. We have the three carbon atoms over here. Yeah, we have the three carbon atoms, right? And if we have the aldehyde group at the first carbon, it is glyceraldehyde. And what will be the keto derivative, guys? What will be the keto derivative? Anyone? Yes. What will be the keto derivative? So it is glyceraldehyde. Yes, glyceraldehyde. And what will be the keto derivative, guys? It is, yes, it is nothing but the di. Yes, someone answered. It is Muskan only. Very good, beta. Very good. It is the dihydroxyacetone. It is dihydroxyacetone. So we have the glyceraldehyde part and the dihydroxyacetone. Now coming on to the four carbon atoms. So this is called as erythrose. This is called as erythrose, right? This is called as erythrose. And yeah, in the ketosis, it is called as erythrulose. Erythrulose, right? Erythrulose. Be very clear with the spelling, guys. Aldehyde derivative of four carbon atoms, it is erythrose. And keto derivative of four carbon atom, it is erythrulose, right? So just the names, guys, right? Now, in the case of pentosis, we have three parts, right? In the case of pentosis, because the functional group can be present at three different parts, So we have ribose, three different carbon atoms, right? So it can be ribose, it can be xylose, and it can be arabinose. 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 Now, guys, for the ribose part, it will be ribulose, right? It will be ribulose. For the xylose part, it will be xylulose, xylulose, right? For arabinose, we don't. Okay, sorry. So yeah, over here only, just you have to remember this is, yeah, the yes, Muskan, very good. For, yeah, for the aldoses part, we have ribose, xylose, and arabinose. And for the ketosis part, it is ribulose and xylulose. For arabinose, we don't have any keto derivative, right? We don't have any keto derivatives. So these are, yes, aldehyde derivative, aldoses, and these are keto derivatives, right? These are keto derivatives, right? For hexoses, guys, for hexoses, we have, okay, can anyone tell me the keto derivative, the commonest example of six carbon atom? Yes, the commonest example, yes, Muskan, very good, very good. Glucose. Can anyone tell me the other examples of this aldoses? Glucose, this you already know. We have galactose, we have mannose, right? We have glucose, galactose, and
Mm.
mannose. These are all aldehyde derivatives, guys, right? And yeah,
Barrels.
yes, Sohail, you're right. But I would request if you just type in the chat box, it'd be more accessible. No, and in the ketosis part, Peter, you already know we have only one part that is called as fructose. Now, when I say fructose, guys, if I say it is a keto derivative, fructose is a keto derivative, then which functional group will be present? I mean, what I've already told you the functional group. Yeah, it is C double bond O, right? Glucose, it is aldehyde derivative. Galactose also, mannose also. Basically, the aldehyde functional group is basically present on first carbon or yeah, the symmetrical chain actually matters, right? We'll draw the diagrams for all these, so no need to worry. How are they different? We'll just come to that part also, right? So that is something you guys have to remember. So this is something very basic. Let us just come to the portion from the Oracle notes, and that as they have like mentioned it. So triosis, basically, you already know it is glyceraldehyde and dihydroxyacetone, right? So they are found in all cells as phosphate, basically the glyceraldehyde part, the dihydroxyacetone part, okay? And one important topic that I'm going to discuss tomorrow, it is glycolysis. So I will be focusing on, yeah, this, yeah, we will again study these compounds tomorrow, again in the glycolysis, the glyceraldehyde part, the dihydroxyacetone part tomorrow in the glycolysis. So these are intermediate no products which are no there in the glycolysis. Tetrosis, basically they are widespread. Okay, we have already discussed about erythrose, right? So again, it's an important part in the carbohydrate metabolism that we'll be discussing tomorrow, right? Again, the pentosis, right? Now, when I say ribose, guys, so ribose, you are very much aware of the fact that these ribose are constituent of RNA and nucleotides. Okay, the deoxyribose, they are basically the constituent of DNA, right? Ribulose, basically, it is produced during the metabolism. I'll be talking about all the pathways, right? So xylose, it is a constituent of glycoproteins and gums. Xylulose, it is a intermediate in uronic acid pathway. Uh, Lysose is basically in the heart muscle. So yes, right? So the biochemical importance is basically for the structure of RNA nucleotide. Basically, they are required for the structure of formation of DNA. They are required. Okay. HMP pathway, we will be discussing the hexose monophosphate shunt, right? It is involved in the function of the glycoproteins, right? So yes. So it is excreted in urine in essential pentosuria. Okay. So we are going to discuss all this, right? Now let us just discuss about the glucose wala part. Okay. The glucose, galactose, and mannose, right? Mm. Now coming on to the glucose, guys, you already know it is the most prominent sugar. Remember, guys, it is the most prominent sugar. It is the most prominent sugar in the human body. In the human body. Okay. Remember, guys, that it is the main source of metabolic fuels. It is the main source of metabolic fuels of mammals, right? Basically, we get the energy, right? It is the universal fuel for fetus. Now, when I say fetus, guys, okay, you already know I'm talking about when the conceptus is in the form of a fetus. Okay, firstly it is zygote, then it is the embryo, then it is the fetus, right? So it is the only source for energy for the fetus. It is glucose. So it is the universal fuel for fetus, right? And glucose is dextrorotatory, guys. The glucose, it is dextrorotatory. Okay, hence otherwise it is called as dextrose. Hence, otherwise, it is called as dextrose. So glucose is dextrorotatory, hence, otherwise, it is called as dextrose also, right? So now coming on to the structure of the glucose, guys. Now, how many carbon atoms are there in the glucose? Anyone can tell me? Yes. How many carbon atoms are there in the glucose, guys? Chalo. Anyone? Yes. We have 6 carbon atoms, guys, right? We have 6 carbon atoms. Now, you already know. Let us just try to understand this. We have the 6 carbon atoms. Okay, we have 6 carbon atoms, right? And you already know glucose is an aldehyde derivative. So the aldehyde group will be present at the first carbon only, right? The aldehyde group will be present at the first carbon only. So the glucose is an aldehyde derivative. So the aldehyde group is always present at first carbon, right? Now, on the third carbon, okay. Now, all the OH are present along this, yes, the right side, right? Except for the third carbon, guys. Except for the third carbon, all the OH you will see over here, okay? Except for the third carbon, the OH is present on the left-hand side, right? Now to balance, right? All the H is like same. Over here, it is CH2OH, right? Yes. So that is something that is the linear structure of the glucose, guys. That is the linear structure of glucose. Linear structure of glucose, right? That is the linear structure of glucose. Now, this is something that we have to remember, right? Hmm. I hope this is clear, everyone, right? I hope this is clear. Now, if we just try to draw a ring structure, guys, I'll be drawing the ring structure for one of the compounds. Later on, you can just draw for yourself as well for other things, right? So if we just try to create a bond between the first carbon atom and the fifth carbon atom, guys, the first carbon atom and the fifth carbon atom, the first carbon atom and the fifth carbon atom. Now, you already know. Now, what will happen? We have this OH group, right? And we have this O group. So this O group will be shared between the first carbon and the fifth carbon, and this OH will go to the first carbon. ये जो OH है carbon वाला, it will again move on to the first carbon, and this oxygen will be shared between the first and the fifth carbon. So this OH automatically moves on to the first carbon, right? I'm drawing a trans structure, guys, right? Before the ring structure, and this oxygen will be shared between the first and the fifth carbon, right? So let us suppose this is the first carbon, second, third, fourth, and fifth. And sixth, again, it is same. So just try to mark the numberings as well. Very, very important it is, right? So this is like this structure. Yeah. Again, the ring structure, guys. So remember, we have it in the form of a pyranose ring. Pyranose ring, guys. So remember, it is a 6-membered ring. It is a 6-membered ring, guys, right? So we have 5 carbon atoms, like plus 1 oxygen. We have 5 carbon atoms and 1 oxygen. So let us start with the carbon fifth. So you already know, let us suppose put this carbon fifth over here, right? To this carbon fifth, you already know we have one oxygen which is joining, right? We have one oxygen, okay? Now this oxygen, it is going to the first carbon. So let me just draw the first carbon also over here. So this carbon, it is joined to the first carbon with the help of this oxygen. Then after that, if you come to the first carbon, then go to the second carbon, right? We are going to the second carbon. Then we have third carbon, okay? Then we have fourth carbon and automatically fifth, right? Automatically fifth. And to the fifth carbon, you already know we have this CH2OH, it is attached, right? CH2OH, it is attached. So this is nothing but the pyranose ring. Okay, it is a 6-membered ring, okay, with 1 oxygen, 5 carbon atoms, right, with 1 oxygen, right? So automatically, guys, you should remember that if I just try to compile it, so you already know we have this kind of a ring, right? So if I just remove the carbon atoms just to show the clear structure, it looks like this. It looks like this. So this is CH2OH. Now at the first carbon beta, the OH is, okay, the OH is again present. So yes, it will be down, and then H is over here. Then at the second carbon, OH is on the right side, so it is down, then H. But on the third carbon, guys, remember the OH is on the left side, so it is up and like this. Rest all the things remains the same, right? So it is OH and H. Yes, like this. So I hope this is clear. I hope this is clear to everyone, guys. Just give me a thumbs up in the chat box if this is clear to everyone. So this is how you have to make the yeah structures, right? This is how you have to make the structure. Doctor, I hope it is clear to you. So yes, now coming on to the next part, that is the galactose, guys. So galactose, the medical importance that you have to remember, guys, it is a constituent of lactose. It is a constituent of lactose, right? Now, when I say lactose, guys, you already know it is called as the milk sugar. It is called as the milk sugar. And remember, guys, this galactose, it is synthesized in the mammary gland. Important. It is synthesized in the mammary gland. Important. It is synthesized in the mammary gland, right? So these are two important parts that you have to focus on. So it is a constituent of lactose, that is the milk sugar, and it is synthesized in the mammary gland. It is synthesized in the mammary gland, guys, right? Now, when I say galactose, guys, now what is the difference between Okay, let me just simply show you what is the difference between glucose and galactose. It is just in the orientation of OH and this. Obviously, galactose is also an aldehyde derivative, so the functional group will be present at first carbon only. Okay, but so remember, it is just similarly like, okay, it is just similar to glucose. It is similar to glucose. So just you have to remember the H group on the third carbon, it is in the glucose, it is on the right hand side. Even okay, on the fourth carbon, it is on the right hand side, and even on the third carbon, it is on the right hand side. So this is the difference between glucose and galactose. Otherwise, the entire structure remains the same. So just a homework for you people, you have to just draw the ring structures for this also, right? Coming on to the next one, that is the fructose, guys. So fructose, I have already told you, it is a ketohexose, right? It is a ketohexose, guys, right? Now, you already know when I say ketohexose, it means it is having 6 carbon atoms. It is having 6 carbon atoms. 1, 2, 3, 4, 5, and 6. And guys, you already know the functional group, it is always present at the second carbon atom. Otherwise, the entire structure is same like glucose. The H is present on the right side. Otherwise, the OH is all present over here, right? So this is the structure of, yes, this is the structure of fructose, right? This is the structure of fructose. So just try to draw the ring structures for fructose and galactose. So I hope this is clear to everyone. So I hope all the monosaccharides are clear to everyone. Just give me a thumbs up, guys, if the monosaccharides are clear to every one of you. Yeah. I hope, Kavita, you are able to see the screen, right? I hope you are all able to see the screen over here, right? Chalo, I hope you all are able to see the screen over here. Chalo. Now coming on to the disaccharides, guys, coming on to the disaccharides. Now when I say disaccharides, guys, you already know disaccharides means two potential sugar units, right? Kavita, yes, very good, beta. So yeah, disaccharides basically means two potential sugar units. Okay, so we have two potential sugar units, right? Two potential sugar units. So how the disaccharides are formed, guys? By joining two monosaccharides together, right? So they are formed by joining the two monosaccharides together. The two monosaccharides together, okay? Now these two monosaccharides, they are joined by a bond which is named as glycosidic bond, okay, which is known as the glycosidic bond. Now, this glycosidic bond is nothing but the covalent bond, okay, the bond which is formed by sharing. It is called as the covalent bond, right? So it is the covalent bond, that is the glycosidic bond. So remember, guys, the disaccharides are basically classified into two types, right? The disaccharides are classified into two types. We have reducing disaccharide. We have reducing disaccharide and non-reducing disaccharide. So we have reducing disaccharide and we have non-reducing disaccharide. Now, when I say reducing disaccharide, what is the meaning of the term reducing, guys? That we have the functional group which is free, right? So in the reducing part, you just have to remember, guys, that at least one free functional group should be there. free functional group should be there. Now, when I say free functional group beta, it clearly means that this functional group is not participating in any bond. It is free, right? कि कोई भी bond नहीं बनाता, it is automatically free. So you guys already know the functional groups are neither but they are the aldehyde group, right? Or they are the keto group. दोनों में से कोई एक free होना चाहिए, right? So either it should be free or either it should be free. So the functional group is the one which makes it reducing, which is the free functional group, right? So that is the meaning of reducing disaccharide, right? Now, when I say non-reducing disaccharides, basically, so a non-reducing disaccharide is the one in which the two functional groups are joined together. I will show you with the help of a diagram also, the schematic diagram. So just remember, these are the two functional groups which are joined together, right? Now over here, the functional groups are engaged in the bonding, right? They are engaged into the linkage, and that is nothing but the glycosidic linkage. They are not free. They're not present freely. They are joined together, and that linkage, guys, we have already, I've already told you, it is the glycosidic linkage, right? So in the non-reducing disaccharide, there is no free functional group. There is no free functional group. There is no free functional group. Now, let us just try to understand the difference between the reducing and the non-reducing with the help of some schematic diagrams. So now again, guys, this is a portion from the notes itself. So they have clearly know they have given so many parts in a very beautiful manner. So this is again a tabular form where all the disaccharides are present: the maltose, isomaltose, trehalose, sucrose, and lactose. So now when I say maltose, guys, so you can very well see maltose is a disaccharide. It is formed of two sugar units. So two glucose are joining together. Okay, the two glucose are joining together. So and they are joining together with the help of alpha 1,4 linkage. Now let me show you why it is a reducing sugar. Let me just try to explain this first point over here. Now if we talk about maltose, guys, you already know, let us simply, we already know we have 6 carbon atoms in glucose. So maltose, it is formed by 2 glucose. So we have to draw 2 glucose over here, right? So just try to draw the 2 glucose over here, right? Now, okay, there's the first carbon, second, third, fourth, fifth, and sixth. Now, guys, you already know that in the glucose, basically the functional group, it is present at the first carbon, right? The CHO group in both the case, right? The other ones, basically, it remains the simple glucose that we all know draw. It's just H. Now, over here also, guys, it is again the same, right? Now, at the third carbon, you already know the H is on the right side. So these are the two glucose, guys. Now, when I say alpha 1,4, as mentioned in the slide, alpha 1,4 linkage. So basically the first, okay, it is between the first carbon atom of the first glucose and the fourth carbon atom of the second glucose. Obviously, when they will join together, then only the maltose will be formed, right? So remember, the joining together of the first and this, okay, this is nothing but alpha 1, to 4 linkage, right? Alpha 1 to 4 linkage. This is nothing but the glycosidic linkage. This is nothing but the glycosidic linkage, guys. So this is something that you have to remember. Now, why I say it is a free functional, I mean a reducing sugar, guys. So again, the definition of a reducing sugar, I already told you that there must be a free functional group. So you can see in the second glucose, the aldehyde group, which is present on the first carbon atom in the second glucose, it is not participating in the bonding. It is free. That's why beta, it is a reducing sugar. It is a reducing sugar. So again, coming to the definition means it, this sugar has to have at least one free functional group, and that is automatically present at the first carbon atom, right? So this is something called as the maltose. According to this particular flow chart, guys, you can very well see it as a reducing sugar. Now coming on to isomaltose, guys, again the same diagram. Now it is glucose plus glucose only. Now what is the difference between maltose and isomaltose? We have alpha 1 to 6 linkage. Alpha 1 to 6 linkage. Now come on to the diagram, guys. So you can very well see it is between the first carbon atom, right? It is between the first carbon atom and the sixth carbon atom, right? So automatically the water molecule will be lost, right? And they both will combine with each other. So it is nothing but alpha 1 to 6 linkage. First carbon atom of the first glucose and sixth carbon atom of the second glucose. So it is alpha 1 to 6 linkage, guys. It is alpha 1 to 6 linkage. So that is nothing but the isomaltose, and alpha 1 to 4 linkage, it is maltose. So they both are reducing sugar, reducing disaccharides, because they have one free functional group in the second glucose, right? So I hope this is clear to everyone, right? Just give me a thumbs up, guys, if it is clear to everyone. Yeah. So yes, that is the isomaltose. Now coming on to the next structure, guys, let us first discuss about the lactose, right? Okay. If you want, you can just no, guys, make. Okay. Now, okay, let us just try to discuss tree halos. Tree halos, it is between the glucose and glucose. Now, guys, you can very well see over here in this particular diagram, if we just take the first carbon atom, right? If we just take the first carbon atom, right? Yeah, I'm just trying to take the first carbon atom of both the glucose. Now it is alpha 1 to 1 linkage. It is alpha 1 to 1 linkage. Right, guys? It is alpha 1 to 1. The first carbon atom of the first glucose, and the second glucose, we are also taking the first carbon atom. Obviously, they both are joining together. They both are joining together. It is alpha 1 to 1 linkage. And guys, over here, no free functional group is present. No free functional group is present. That's why it is called as the non-reducing. Okay, the tree halos. It is a non-reducing disaccharide. It is a non-reducing disaccharide or a non-reducing sugar, right? Because there is no free functional group, right? So tree halos is also done. Now coming on to the lactose, right? Okay, let us first discuss the lactose. Lactose is galactose plus glucose. Lactose is galactose plus glucose. So you already know, guys, we have, yeah, the 6 carbon atoms, right? Let us suppose this is the galactose and we have glucose, right? So you already know, yeah, in the third carbon atom and the fourth carbon atom, over here, it is only the third carbon atom which is having H over here. Rest the entire structures remains the same. So it is glucose, right? And it is galactose. Now, when I say lactose, it is between the first carbon atom of the galactose and the fourth carbon atom of the glucose. So there is a linkage between the first and the fourth carbon atom. Now see, these both structures are different. The first one, it is galactose, and the second one, it is glucose. That's why it is beta 1 to 4 linkage. It is beta 1 to 4 linkage, guys. It is beta 1 to 4 linkage, okay, between the first and this the second glucose wala structure, right? So it is the beta 1 to 4 linkage. Again, guys, why lactose is a reducing sugar? Because we have a free functional group, free aldehyde group that is present in the glucose. It is not participating in the bonding. Bonding में कौन-कौन participate कर रहा है? It is the first carbon atom of the galactose, that is the aldehyde group, and the fourth carbon atom in the glucose wala structure. So the second glucose wala structure, you can see we have a free aldehyde group. That's why it is a reducing sugar. It is a reducing sugar. So that is something you guys have to remember. So we have lactose over here. Okay. Again, coming on to the sucrose. Sucrose beta, it is non-reducing. Sucrose, it is non-reducing. It is between glucose and fructose. Okay. It is between glucose and fructose. Right. It is between glucose and fructose. So you guys already know that if we just try to draw the glucose, guys, we have 6 carbon atoms. And yes, just I'm trying to draw very simple, guys, over here. So this is glucose. And yes, now yeah. So we have this glucose and we have fructose, right? So fructose again is a 6 carbon compound and it is a keto derivative. So it is present at second carbon, the keto group, it is fructose. So when I say alpha 1 to 2 linkage, guys, it is between the first carbon atom, means the aldehyde group of the glucose, it is bonding with the keto group of the fructose. So obviously, if both the functional groups are involved in the linkage, there is no free functional group. There is no free functional group. That's why sucrose, it is a non-reducing sugar. That's why sucrose, it is a non-reducing sugar. क्योंकि कोई free functional group है ही नहीं. ना first glucose के structure में है, and in the case of the second also, the fructose, we don't have any free functional group. So that is something you guys have to remember. So again, just trying to summarize, guys, we have all discussed these important points that has been mentioned in your notes. So just trying to know examples. You already know we have maltose, isomaltose. Okay, we have lactose and we have lactulose. Lactulose, right? So maltose is nothing but, okay, what are the monomer units? Now, name of the disaccharide. What are the monomer units? Now, when I say monomer units, maltose is formed from glucose plus glucose. Okay, it is formed from glucose plus glucose. Isomaltose, it is also formed from glucose plus glucose. Lactose, it is galactose plus glucose. And lactulose, it is galactose plus fructose. Now, these are the monomer units, and accordingly, guys, you should also know the linkage wala part, the linkage, right? So, in the first one, we have alpha 1 to 4 glycosidic linkage. Glycosidic linkage. So, in the second one, we have alpha 1 to 6. Then we have we have beta 1 to 4 glycosidic linkage, right? So again, this is no linkages over present over here. Yeah. So lactose synthetically derivative, right? So that is examples of reducing disaccharides, right? Now coming on to the non-reducing one. So you have sucrose, right? We have sucrose. And the second one you already know we have trehalose. Sucrose, basically it is glucose plus fructose, right? It is alpha 1 to 2 linkage, right? Alpha 1 to beta 2 actually. We have two different compounds over here now. So it's just like this. It is alpha 1 to beta 2 linkage. Alpha basically for the glucose and fructose is for the, yeah. And trehalose is nothing but glucose plus glucose. Now, when I say glucose plus glucose, it is the same structure. It is alpha 1 to 1 linkage. We have already discussed this. So these are the examples of reducing and non-reducing disaccharides that you guys should know, right? Okay, perfect. Now coming on to the polysaccharides, guys. So we have already completed the definition. We have already completed the monosaccharides wala part. We have already completed the disaccharides with along with all the diagrams, the schematic representations, and the biological importance, right? Now coming on to the polysaccharides, guys. You already know the polysaccharides. Basically, we are having more than 10 sugar units, right? We have more than 10 sugar units. Now, this polysaccharides, they are classified into two categories, guys. They are classified into two categories. We have homopolysaccharides, and we have heteropolysaccharides. Heteropolysaccharides, right? Now, guys, you already know when we say homo, guys, homo means same, right? Poly means many sugar units. So remember, yeah, we have one type of sugar unit which is repeating. That's why poly, right? So we have one type of sugar unit or the monomer units. We have one type of sugar or the monomer units. The heteropolysaccharides, they are also called as heteroglycans. They are also called as heteroglycans. Basically, we have different or guys, more than one type of monomer units. Monomer units. Okay. So the polysaccharides, they are having more than 10 sugar units. The simplest definition, poly means many, saccharides means sugar. They are classified into homopolysaccharides and hetero. Homo means one type, one similar type of sugar unit. Hetero means different type or like more than one type of monomer units, right? The sugar units. Now, the examples of these homopolysaccharides, guys, you should remember we have glycogen, right? We have glycogen, starch, cellulose, right? Inulin, chitin. So we will be discussing it one by one. We have glycogen, starch, cellulose, inulin, and chitin. Heteroglycans are basically the glycosaminoglycans. Glycosaminoglycans, okay? They are commonly glycosaminoglycans. They are called as GAGs, right? We have something called as agarose. It is also a heteroglycan. Again, we have something called as pectin. So we'll be discussing it one by one. So as of now, just try to make a flowchart in a very simple manner. Like, so this is the classification of polysaccharides done. So yes, so we have to study this one by one. Now coming on to the next one, guys. So we'll be discussing about the starch, glycogen, and cellulose. Again, guys, the picture is taken from the Auricle notes. All the important pictures along with the notes part, they are like in a very typically they are very good, right? So just try to follow the notes as well along with the which one I'm writing along with you, right? Just try to correlate with the notes given in the Oracle. So they have beautifully drawn all the schematic diagrams, right? Now coming on to the glycogen, guys. Now glycogen is something which is made up of glucose, right? Excess glucose is stored as glycogen in the human body, right? So remember, guys, that the glycogen, it is made up of alpha D glucose. I'll be coming to this D and L wala term tomorrow. Okay. So as of now, remember it is made up of glucose simply. Okay. Now all the glucose units are joining together to form the glycogen. So I can simply say that it is a branched polymer. It is a branched polymer. Okay. Obviously, the glucose is combining with each other like this to form the glycogen, right? So it is just like this. Okay, so yes. Again, the point that I have already discussed, that it is the storage form of carbohydrates. It is the storage form of carbohydrates in animals. It is the storage form of carbohydrates in animals. So it is also known as the animal starch, right? It is also known as animal starch, right? Now, very important, two important points that I will be mention. No, I'll try to mention over here on the screen, that this glycogen, guys, it is stored in two different sites, right? It is stored in two sites. Okay, can anyone tell me one of the sites where it is stored, guys? Anyone who would just like to tell me where this particular thing is stored? Yes. Firstly, guys, yes, it is the muscle where it is stored, right? If someone has answered, Ishika, very good, very good. It is the liver and the muscle. Very good, Peter. It is the muscle and the liver, guys. It is the muscle and the liver, right? So we have the liver where it is stored, and we also have the muscles where it is stored. So we have the liver, and yes, we already know we have the muscle wala part also, right? So it is stored in the liver and muscles. So now if I just try to simply draw the structure of the glycogen, guys, so let us just try to summarize that this glycogen, it is formed from the glucose, right? It is formed from the glucose, right? Glucose and glucose are joining, right? Now, when glucose and glucose are joining together with each other, it is called as alpha 1,4 linkage. Okay, the first aldehyde group of this glucose will be joining with the fourth carbon atom of the second glucose. So it is alpha 1,4 linkage, right? Now, if I told you it's a branched polymer, it's a branched polymer. So let us just try to show the branching over here also. So these are again the glucose, guys. These are again the glucose, which you can see over here, right? Now you can see that, okay, this glucose and this glucose at the branching site, beta, at the branching site, right, where the branch is formed, at branches. Now, what is the linkage? They are joining with the first carbon atom of this glucose. The aldehyde group will be joining with the sixth carbon atom of the other glucose, which is at present at the branching point. So we have alpha 1 to 6 linkage. Very, very important. Alpha 1 to 6 linkage. Over here also, yes, we will have alpha 1 to 6 linkage. Yes, Ishika, you are right. Yes, Ishika, you are right. So over here also we will have alpha 1 to 6 linkage. Again, but between the glucose and the glucose, again it is same, that is alpha 1 to 4. So we have alpha 1 to 4 linkage in the linear structures, and at branching points we have alpha 1 to 6, right? Now, two questions that have been asked in the exam. That is something you guys have to remember over here. That is the total content of glucose. Okay, remember it is more in the muscle. Okay, it is more in the muscle. But if we just try to see the percentage by tissue volume, okay, just by comparing it by percentage by tissue volume, the glycogen is more in the liver. Glycogen is more in the liver. So in the liver, guys, you just have to remember the glycogen is stored as aggregates of beta particle, right? It is stored as aggregates of beta particle. This is nothing but the rosette of the beta particles, right? Okay, and in the muscles, the single beta particle has 60,000 glucose units, right? So we have the beta particles over here, right? And these beta particles is having 60,000 glucose units. Glucose units. right? So that is about the glycogen. That is about the glycogen, guys, right? So in a very simpler manner, I've just tried to draw the structure, the important points that are relevant for the exam. So these points you can't um, forget to miss all this, right? Now, so this is about the glycogen. I hope you can just see the structures of the glycogen over here, right? So we have the branching points where no the alpha 1 to 6 linkage is present, but in the linear structures, guys, remember we have alpha 1 to 4 linkage, right? So now coming on to the starch, guys. So remember that the starch, it is the storage form. It is the storage form of carbohydrates in plants. Okay, it is the storage form of carbohydrates in plants, right? And basically they are of two types that you have to remember. We have amylase and amylopectin. Amylopectin. So the amylase one, guys, it is soluble in water, right? Whereas the amylopectin, it is insoluble in water, right? The amylase, it is unbranched. The amylopectin, it is branched, right? So it is just like amylase is just like this. It is unbranched structure. So yes, all the glucose units are arranged like this only. Let me just try to draw it. Yes. So they are arranged like this only. Whereas amylopectin, it is branched, so it is like this only, right? So that is the simplest difference between the amylase and the amylopectin, right? So amylase being soluble and unbranched, amylopectin being insoluble and branched, insoluble and branched. Now coming on to the next one, that is the cellulose. Remember, guys, cellulose, it is the major dietary fiber. It is the major dietary fiber. Okay, it is composed of the beta D glucose, and it is having, yes, the beta 1 to 4 linkage. Okay, we have beta 1 to 4 linkage. Now, why we can't digest no the plants? Because the humans basically lacks enzyme. Okay, the humans lacks enzyme to hydrolyze this particular, yes, to hydrolyze beta 1 to 4 linkage, right? And yes, the cellulose. So yes, that is something you have to remember. So the human lacks enzyme to hydrolyze basically this beta 1 to 4 linkage, right? Coming on to the next one, that is the chitin, guys. Two important points that you have to remember, that this chitin is found in the exoskeleton of insects. It is found in the exoskeleton of insects, right? And it is composed of N-acetyl glucosamine, right? It is composed of N-acetyl glucosamine, NAG. We have the acetyl group, glucose, and the amine group, right? Now, coming on to the next one, that is the inulin, guys. Inulin, basically, it is a homopolysaccharide, right? It is homopolysaccharide. We have already discussed this, right? It is a homopolysaccharide. It is polymer of, yes, very good, Peter. It is polymer of beta D fructose, right? It is polymer of beta D fructose. It is also called as fructosin, right? Right. Now, important point is it is used as an external agent. It is used as an external agent to check for the GFR. Now, GFR stands for the glomerular filtration rate. Okay, you'll study this in physiology, right? And this test, beta, it is known as the inulin clearance test. Inulin clearance test. So this is used as an external agent to check for the GFR, and this test is called as the inulin clearance test. Now, that is the important points that you guys have to remember. Yeah. See, this external agent basically means we are actually giving it from outside, guys, right? So it's not something which is present. Yeah, we are adding this external agent into our body just to check for the GFR. ठीक है, Ishika? Coming on to the last one, that is the dextran and the dextrin part. So remember, guys, the dextran, it is nothing but the homopolysaccharide of glucose. The homopolysaccharide of glucose, right? And yes, the dextrin. Dextrin is nothing but the hydrolytic product of starch. Hydrolytic product of starch. So that is the simplest difference that you guys have to remember. The homopolysaccharide of glucose, it is called as dextrin, and dextrin, it is basically hydrolytic product of starch. Even dextran, you will be studying in physiology. It is related to the plasma volume expander, right? So that is the point that we will be discussing it in physiology. Coming on to the last one, that is the pectin beta. So the pectin, basically, it is considered as the dietary fiber, right? It is considered as the dietary fiber, right? Pectin, it is a heteropolysaccharide. I have already discussed you, it is not homo, it is heteropolysaccharide. Okay, so it is okay. Now, if just a composition part is asked, so it is made up of galactouronic acid, right? And plus it is made up of arabinose also. Arabinose we have just discussed right now, right? So galactouronic acid, it is basically present in majority wala part. So that's why pectin is also considered as a heteropolysaccharide, because we have different monomer units. We have galactouronic acid and arabinose also, right? So yes, that is about the all the important points related to this. Yes, the homopolysaccharides. So guys, we'll just try to stop it over here. The hetero part will be focusing it tomorrow in the upcoming first 10 minutes of the lecture tomorrow, right? So we have already discussed regarding the monosaccharides, the disaccharides, reducing and non-reducing, how carbohydrates are formed, right? And yes, the important structures like glycogen, starch, cellulose, chitin, inulin, dextran, and pectin. So yes, we'll just try to meet up tomorrow, guys, right? So if there is any doubt, you guys can text me, right? So yeah, I'll just try to end the session now. Okay. We'll be having MCQ questions tomorrow, guys, in my lecture, because today I've just discussed some theory part. So tomorrow we'll be discussing some important MCQs, right? Sure. Okay. So I'll be okay, Shikat. Welcome. So I hope you understood. So first, shall I like stop it? Shall I leave the session or yeah? Okay.
Yes.
Hmm. Sure. ठीक है sir.
Thank you, sir. Thank you.
Okay.
Yes, sir.